Okay, so let's look here at DNA just a bit. Now, again, there's a nice little video here about the structure of DNA and what it does, and I'll put that on Classroom. But DNA itself stands for deoxyribo, that's the sugar, nucleic acid. So this is something that pretty much you're going to encounter wherever. So here's the structure of a nucleotide. A nucleotide is made of a phosphate group, which is this phosphorus and oxygen, connected to, by a covalent bond, this deoxyribose sugar. These two parts make up what's called the backbone of a nucleotide. So if you were looking at this in terms of a ladder, this would be the sides of the ladder. And then the sugar is connected to one of the 4 nitrogen bases. And the sugar and the nitrogen bases may vary a bit if you're talking about DNA or if you're talking about RNA. But we're in DNA right now, so we're talking about deoxyribose sugar, and we're talking about A's, T's, C's, and G's. Now, I love memory devices, so I'm going to provide you with some memory devices when it comes to the nitrogen-base pairings of your DNA. I also have a little device here for remembering which one is which in terms of a purine or pyrimidine. So here are your 4 nitrogen bases or your nitrogenous bases. And adenine and guanine are called purines, and cytosine and thymine are called pyrimidines. So this you'll definitely encounter again in higher level science classes. So an easy way to remember which one is which is purag, P-U-R-A-G. So if you say purag, that means that the purines are adenine and guanine. And then that would leave cytosine and thymine for the pyrimidines. Now that's not my personal memory device. I just snagged that one from some study group that I found on the internet. But the way that I always remembered which one is which, purine, smaller word, but it's the bigger molecule. Pyrimidine, bigger word, but the smaller molecule. And that's an easy way to remember a lot of things. That's an easy way to remember acids and bases, and that's an easy way to remember surface area to volume ratio and the size of a molecule is by using opposite day. Now, when it comes to who pairs with who in DNA, we can use this little gem right here. All turkeys can gobble. So A pairs with T. They're complementary or complementary bases. And then C pairs with G. Now we're going to talk about making copies of DNA. So we know that at certain points in a cell's life, DNA has to be replicated. This process of replication is going to occur before mitosis. It's going to occur before meiosis, during interphase, specifically the S phase of interphase. Now, the process itself is called replication, and the goal is to make 2 identical copies of the DNA. So here we have the original, and then here we have the 2 copies. Now, this process is going to occur in the nucleus of a eukaryotic cell, like ourselves, and in the cytoplasm of a prokaryote. And every time this happens, you end up getting a new piece of DNA that has an old strand and a new strand. So this old strand is actually called the parent strand and serves as a template to make the new strand, which is called the complementary strand. Because again, we're talking about complementary bases. So when we go to do this, it's very simple. You just have to remember your nitrogen-based rulings, A with T, C with G. Now, if you look at this, though, you can see some numbers, 5s and 3s and 3s and 5s. So this all has to do with what's considered top and bottom. So the DNA molecule is called anti-parallel. So if you look, one strand is going to be right side up, the other strand is upside down. That's the way it works. Something else about this 5 and this 3, all that has to do with is positioning of the molecule. So which carbon is at the top. Is it the 3 or is it the 5? And that's where you get these numbers from. So you'd probably deal a little bit more with that in biochemistry. So let's replicate this one. So all I have to remember, all turkeys can gobble. So C, can gobble. C will go with G. G will go with C. T will pair with A. A will pair with T. A will pair with T. G with C. C with G. G with C. G with C. C with G, and T with A. So it's that easy. Now, what keeps them together, and you can see like these little dashed lines right there. So at this point, they have to be unzipped to separate the two strands, but when you go to form them, these will get longer, they'll connect together. The bonds that hold the two strands of DNA together are called hydrogen bonds. And the hydrogen bonds are what are responsible, or here they are, there, the hydrogen bonds. They're responsible for the twist in the DNA. So here's another one from practice, so you could see what you could do with this one. Remembering all turkeys can gobble. And this should be what you have. C with G, A with T, G with C. And then what we would have in here would be the hydrogen bonds. And replication is that easy. So then this piece of DNA will go on to maybe be in the daughter cell. So if you're making some new skin cells in mitosis, you would need a copy of the DNA. You don't donate, you have to give a copy. Now this process works out pretty well. There is an enzyme, DNA polymerase, that's responsible for putting the bases together. It's also responsible for proofreading. So it's kind of like a couple of jobs there, putting together and make sure that it's putting the correct bases together or the complementary bases together.